Today is the first day of fall. So Scott and Sharon, welcome to the first day of fall and welcome to the Oppenheimer Fall Healthcare Conference. And please take it away.
Thank you, Hartaj, and thank you to your firm for hosting us. I'm Scott Fine. I'm the CEO of Cyclo Therapeutics. We're going to leave time for Q&A. The most important part of our presentation, in my view, is the medicine and science, which Sharon will cover. So I'm going to give a quick introduction, and then we'll pass to Sharon so she can walk you through our clinical programs and our science. Everyone has seen this before. We have a deep expertise in cyclodextrins, which are an elegant little sugar molecule that has an affinity towards the cholesterol, hence it has a benefit in treating Niemann-Pick type C patients. We've got over 10 years of utilization of Trapsol Cyclo, a beta cyclodextrin for the treatment of Niemann-Pick type C through compassionate programs. This compelled us to stand up the phase 1 and phase 1/2 clinical trials in Europe and in the United States, which have led us to our Transport NPC clinical phase 3 enrolling clinical trial that we are currently standing up right now for Niemann-Pick type C. We also have an Alzheimer's program that Sharon will talk about, where we have an N of 1 and very strong data, and we've been in front of the FDA and expect that we will be in front of the FDA before year end, with an anticipation of getting approval to stand up a phase 2B clinical program by year end. Very importantly, as we prepare for commercialization, we've got 60-month stability on our proprietary state-of-the-art formulation of Trapsol Cyclo beta cyclodextrin. This is very key with respect to as we move into commercialization and look at not only maintaining and managing our clinical program, but also as we prepare for commercialization and launch upon approval. We're a late-stage company with a late-stage phase 3 enrolling clinical program for Niemann-Pick type C. And as I'd said, we're also looking to stand up the phase 2B clinical program for Alzheimer's in the second half of this year. We've got a very diversified and strong management team. Jerry Cox, who is our CMO, acting CMO, came from Sanofi Genzyme, where he led the lysosomal storage group there. Russ Belden was at Genentech for 16 years and did our epidemiology work as well as our commercialization work as we're preparing to launch commercially our programs around the world. Mike Lishak is our head of regulatory, also Sanofi Genzyme, has great rapport with the EMA and FDA, and has truly helped us guide and stand up and working along with Sharon and the clinical team to prepare a robust clinical program in our phase 3. Our Transport NPC program, as we've named it, as I said, is enrolling. It's our phase 3 pivotal program. It's the only available clinical trial available in the world today for patients who are suffering from this terrible disease. And we're moving very quickly to enroll this program so we can get to an interim analysis that Sharon will talk about at 48 weeks based upon our trial design, which we're quite excited about based upon the historical utilization of our drug and what we've seen from our compassionate programs. Symptomology of this disease, and by the way, this disease is sometimes referred to as childhood Alzheimer's, but you have an enlarged liver, severe liver disease, respiratory functions. Many of these patients can't speak. They have seizures. Many of them are ambulatory. There is no U.S. approved drug for Niemann-Pick type C. And in the EU, there is miglustat, which I will talk about with this slide, which is the bullet point number 2, Zavesca, which is known as miglustat. It is used off-label in the United States. It's approved in Europe. As I'd said, we are the only approved clinical program recruiting patients right now. Orphazyme received a CRL in June. They currently still have an expanded access program in works. And Mandos Health acquired Abrobetadex out of the bankruptcy of Mallinckrodt, and they too have an expanded access program. The key to this slide, in my view, is the boxes to the right. As you can see, the competitive landscape is going after the neurological manifestations of the disease. We give our drug through an intravenous route of administration. And thus, we are treating not only the systemic, but also the neurological manifestations of the disease, because we do know our drug is getting into the brain, and we're seeing those effects. And again, Sharon will discuss that in a few seconds. It's an ultra-rare disease, 1 in 100,000 live births. We believe there are 9,000 patients in 80 countries. The highlight here from a commercial standpoint is very key. We see that there's potentially an opportunity of a $500 million revenue market here globally for the treatment of Niemann-Pick type C. I chuckle a little bit because our valuation currently at 40 million in defending a valuation versus a revenue model. I'd be very satisfied with 100 million or 200 million in revenue based upon what I know multiples will trade for in the marketplace. But it is a large market opportunity for us just for Niemann-Pick type C, which of course we wished it weren't for the benefit of the patient population. Sharon, would you please take over?
Great, thanks so much, Scott, and hello, everyone. I'm happy to be with you. I'm Chief Scientific Officer and Senior VP for Medical Affairs here at Cyclo. So I'll walk you through some of our data and bring you up to speed on the main features of our phase 3 trial. To orient you to the top left, the mechanism of action of our drug. So first, as Scott said, the defect in Niemann-Pick disease is that cholesterol gets stuck in cells. And when it gets stuck in cells, the cells don't function normally. That leads to the manifestations of the disease. The cholesterol stuck in cells is shown actually in the middle panel there. The reason why it gets stuck is because of faulty NPC1 protein in most cases. And the job of NPC1 is to move cholesterol normally through a compartment of a cell called the lysosome. Over to the right in that top upper left panel is how our drug works in a very simplistic graphic. Essentially, our drug is a donut-shaped molecule which has an affinity for cholesterol in the core. And so when our drug meets this excess cholesterol in cells, it actually is able to move it out of the lysosome and into normal processing. This allows the cell to function normally. All of that data have been learned from animal studies. So what we've done in our programs is replicate some of those kinds of findings. Over to the right is one example of that. In our phase 1 trial, which was a 14-week trial in Niemann-Pick patients age 18 and older, these are liver biopsies. And to the left of each panel, you can see a bright blue in each of those panels at baseline before we presented our drug. The bright blue is a fluorescent marker for cholesterol. So you can see the livers are chock full of cholesterol. After 7 doses or 14 weeks, the tissue is normalized. And again, this shows us that what we expected to see based on animal studies, we're clearing cholesterol out of, in this case, liver cells, which is what we would expect to see. Now, we cannot biopsy the brain, but we can look at biomarkers in the brain, and that's what's shown in the bottom part of this slide. So over to the bottom left, this is a figure of a metabolite of cholesterol called 24S-hydroxycholesterol. This is produced in the brain and exported out of the brain. It can be measured in the CSF, the cerebrospinal fluid, or in serum. We measured it in serum. What you can see there in the graph is 2 distinct peaks, sets of peaks. Over to the left is the first dose in our phase 1 trial, and over to the right is the 7th dose in our phase 1 trial. And basically the sharp incline that you see at the beginning of those peaks, that is us measuring 24S hydroxycholesterol, which is being exported out of the brain after infusion. And so there's a peak we can detect, and then it subsides. After the 7th dose, once again, there's a peak, and then it subsides. And this tells us that we are removing excess cholesterol from the CNS, which is exactly what we would want to see. Over to the right, we can see what happens to tau. And so tau is a protein which is involved in stabilizing neuronal infrastructure, the cytoskeleton, if you will. In neurodegenerative diseases like Niemann-Pick disease, Alzheimer's, Parkinson's, tau is increased in the cerebrospinal fluid. What we see here is, again, our phase 1 study, baseline, and then over to the right at the end of 7 doses. The 200 level or so on the y-axis, this is normal levels of tau. Anything above 1,000 up to 2,000, this is Alzheimer's range of tau in the CSF. And so Niemann-Pick patients in our study, in this phase 1 study, had excessive levels of tau in their CSF. 6 out of 10 patients reduced tau over time. So by the 7th dose, there was either a dramatic reduction or a slight reduction in tau in those patients. Patients who were stabilized or normal levels tend to be stabilized, and 2 patients increased, one slightly and one dramatically. And so the overall trend here is decrease in tau, telling us that we are probably having a neuroprotective benefit, which is very important when we think of the neurologic manifestations of Niemann-Pick. Next slide, please. So here's a summary of where we are and where we're going, and I'll give you some more data slides in just a moment. But I've noted our phase 1 trial, some of the mechanistic findings that we have, and our phase 2 trial showed us signals of efficacy. I'll show you a data slide of that in a moment. But with the favorable safety profile and signals of efficacy that we're seeing from our phase 1 and phase 2 trial sets us up perfectly for our Transport NPC phase 3 trial. And I will note that we had approval from EMA and FDA to proceed with our phase 3 trial even before our phase 2 finished. So we showed all data that we had, and we're given the green light to move ahead with our phase 3. Importantly, I'll note as well that the EMA, the FDA of Europe, asked us to also include patients under the age of 3 for safety purposes, so infants up to age 3, noting that our drug potentially has a preventative effect. And so we'll be able to see that in that cohort of up to 12 patients, because patients in that cohort do not need to be symptomatic. That's very important to us, certainly to the patient community. If we can prevent symptomology, that will be huge to the patient community. You can see our designations here, and I know that Scott will speak about those more toward the end of the presentation. I'll go back to data. So here's some data from our phase 2 trial. Again, this was a 48-week trial. We give our drug intravenously every 2 weeks. We'll focus over to the right. These are the 9 completers. So 9 out of 12 patients completed this 48-week trial. And what you can see here is change in a 5-domain NPC severity scale. So the 5 domains that matter most to patients for their quality of life are the ability to speak, to swallow, to walk, fine motor skills, and cognition. These 5 domains have really become sort of the mainstay of how the clinical research community looks at disease progression in NPC. If we look at our data collected over 48 weeks through the lens of the 5-domain scale, we can see that the overwhelming narrative here is one of stability. So you can see in each of these groups, but certainly over to the completers, 0 is stability. That is no change in the 5-domain score over 48 weeks. Over to the left in each panel, the minus numbers, that's improvement. So patients improved during this time in those 5 domains. And over to the right is worsening. Now, worsening at a level of 1.4 would be the norm based on literature, but we can see patients worsening at less than that. So this tells us that we're having a benefit in terms of less worsening than would be expected. So the overarching message here is one of stability, less worsening, or in some cases, even improvement. We'll go to the next slide, which shows pretty much the same narrative, but different data sets. So the phase 1 trial is a 14-week trial, as I noted. All 8 patients who were eligible for the extension protocol signed up for it. That speaks volumes in our view about their experience during the phase 1, even after 7 doses. But all 8 of 8 moved into the extension protocol, which allows them to receive infusions at home. And so what we see here are data from each patient, from their first experience with the drug in the phase 1 trial to the last data point in the extension protocol. That's why there's variability in these pairs of bars that you see in terms of time in the months that are noted below each set. Over to the left of each bar is what happened to the patient, their actual change in score in that 5-domain scale. and the bright blue in each pair is what would be expected based on a 1.4 per year change in score. And so this, if we look at the first 3 patients starting from the left, those patients progressed in disease, but not as much as would be expected based on the 1.4 per year published literature. One patient, the fourth one, did worsen more than would be expected. The next 3 were all stable. And the last patient over to the far right improved overall. And so again, this underscores the narrative of this is a story of stability, of less worsening than would be expected, or even improvement. We'll go to the next slide. The upper part of this slide basically is a summary of what I've just said from the previous 2 slides. I won't spend a lot of time there, but happy to discuss this during the discussion period. I want to move to the main features of our transport NPC trial, because we've learned a lot along the way from our phase 1 and phase 2, and we've used all that learning as we designed and reached approval with FDA and EMA on our trial. Essentially, the primary endpoint is either the 4-domain scales or the 5-domain scale. Outside of the US, we're using that very same 5-domain scale, which I just referred to in the previous slides. In the US, it's the same scale minus cognition, and that was developed again in close consultation with FDA. We've increased the number of patients for this study. In fact, there are 93 patients needed in the study, which is twice as many as has been used in prior studies. And as Scott has noted, those trials failed in one case, actually in both cases in the US, and still waiting to hear about Orphazyme by the end of the year. We've also included something called the ACES score in our trial. If we can just go back to the last slide. The ACES allows us to bring homogeneity to our patient cohort. And that's important because this is a hypervariable disease. And what we want to do in our phase 3 trial is make the patients look as much as possible like each other so that we can predict their disease progression and see signal between the control group and the intervention group. And then lastly, we have a 2-year trial, which is twice as long as any other trial, but the other trials failed. But importantly, what we're doing is looking at our data at the 1-year mark, and that's because we have seen signals of efficacy, very promising at the 1-year mark from our phase 2 trial, which I just referred to. So we have a 2-year trial looking at the interim at 48 weeks. If by chance we don't see efficacy, we still have the opportunity to continue to collect data up to 2 years. So we have a lot of sites in many countries, and that's because we have an aggressive enrollment strategy. We've selected our dose based on efficacy and safety from our prior trials. And I've said something about our composite score, our primary outcome measure. We have secondary endpoints, which all back up the primary. These will be objective measurements of features of the disease that are reflected in the primary. And then importantly, we are looking at systemic features of the disease, in particular, the liver and the lung function as exploratory measures so that we can get the complete picture of what's going on in patients in our Transport NPC trial. Next slide, please. This is a slide which gives a little bit of the human face of one patient's experience who has been in the extension part of our phase 1 trial. So 15 months of experience with our drug at the point that we put the slide together. The patient was 59 at diagnosis and had ataxia, slurred speech, had very good cognition, but had significant challenge with coughing while eating. Joined the phase 2 after a positive experience with the phase 1. And you can see below the way that the patient describes their experience with the drug. Within hours, speech and swallow improve. And I'll sidebar, swallow is so important. Being able to swallow without coughing in this disease is very important because many patients will pass away from aspiration pneumonia which is related to the swallow function. So this is a real risk factor. And if we can improve swallow in patients, then that's a huge improvement in the quality of life. This patient sees very quickly an improvement, which lasts 5 to 10 days, and then starts to look forward to the next infusion just a few days after that. So you can see the benefits are listed in that slide. I'll move to Alzheimer's disease now. We were asked, well, first let's talk about the similarities. So cognitive decline is a feature of both Niemann-Pick disease, as Scott mentioned earlier, and certainly of Alzheimer's disease. There are also some cellular and pathologic similarities, including amyloid plaques, increases in tau in the CSF, as I've mentioned. We'll go to the next slide. We were asked to provide our drug on a compassionate use basis to a single late onset Alzheimer's patient. This is a geriatric patient. We said yes because of the similarities between Niemann-Pick disease, about which we know quite a lot, and Alzheimer's disease. Plus, there are compelling animal data from the Flint Beal lab at the time from Cornell showing that cyclodextrins given subcutaneously to an Alzheimer's mouse model could not only reduce plaques in the brain and tau immunoreactive areas in the brain, but also, very importantly, improve spatial learning in the mouse model. So we said yes, worked with FDA and the treating physician on a protocol. We provided our drug at relatively low levels, starting at 500 milligrams per kilogram on a monthly basis, again, out of an abundance of caution, given that this was the first geriatric experience with the drug. We did this for 18 months. And over to the right, you can see the mini mental state evaluation scores. Now, 30 is normal. That's at the top. The stippled line is what one would expect from a late onset Alzheimer's patient. Basically declining. Most, if not all, late onset Alzheimer's patients will decline in this assessment tool over time. The patient in this compassionate use study did not decline. In fact, improved for periods and stabilized, as you can see in the green line there. Importantly, the physician and the family and the patient themselves noted less volatility and better word finding ability. And so because of all this, FDA at the 12-month mark allowed us to continue onward. We did this for 18 months. And then we went to FDA and asked for scientific advice on submission of an IND on Alzheimer's. We received a green light to submit. And we do expect, as Scott said earlier, to submit our phase 2B clinical trial by the end of this year. We're very much looking forward to that. I'm going to stop there and pass back over to Scott.
Thanks, Sharon. So as I mentioned, everyone, we have a late phase asset in our phase 3 study for Niemann-Pick type C. Sharon's mentioned where we are with our Alzheimer's program. We're quite excited about where we'll be by the end of the year with the interactions that we're having with FDA. The Transport NPC program is the only actively enrolling clinical program for NPC patients right now. We have orphan drug designation in the US as well as in Europe, and based upon our pediatric interactions with the EMA, we have 12 years of exclusivity under the ODD in Europe. We have filed a patent around our Alzheimer's program. It's currently pending. If issued, it will give us protection through 2039, so we're quite excited about that. We've got multiple value-driving milestones expected by the end of the year. We can't, of course, tell you what they are, but we're expecting them based upon where we are in our corporate and clinical pathways. We've got a very interesting board of directors and a very interactive and involved board of directors. And I'll note, of the $42 million that we've raised since we got involved in standing up these clinical programs in 2014, of the $20 million of this has come from either ourselves or our board or our friends and family. And I'll take note of Patrick Astronic of US Pharmacia. If you look at our holdings, you'll see that Novid LP is a large shareholder. They've invested approximately $10 million into our company. Marcus Seeger is our chairman. He is the CEO of a very large generic and biosimilar company in Eastern Europe called Pol Pharma. And Bill Shanahan, our senior member of our board, is a former president and COO of Colgate-Palmolive. So we've got a deep bench of knowledge in business and a very involved and engaged board of directors that have helped us build out the platforms that we're presenting. Sharon, why don't you quickly talk about the SAB, please?
Sure. Thanks, Scott. So I co-chair the Scientific Advisory Board along with Dr. Rita Colwell, who is one of the most prominent scientists in our country. She's a microbiologist by training, but former head of the National Science Foundation and former and winner of the National Medal of Science. So she's really quite prominent. She readily accepted to co-chair this Scientific Advisory Board with us so that we could look at potential future indications. Benny Liu and Caroline Hastings are the other two noted here. Benny is the gastroenterologist who, as a young postdoc, identified cyclodextrins as being able to clear cholesterol from the Niemann-Pick mouse model. And Caroline Hastings was the first physician in the United States who used cyclodextrins, our drug, in compassionate use, and then shared her protocol globally. These were the data that allowed us to launch our formal clinical programs.
Thanks, Sharon. Thanks, Hartaj.
Great. Scott, thank you, Sharon, so much. And Scott, really, really nice presentation. I've been working about 30 years of my life, Scott. So, when the Washington Nationals won the Major League Baseball championship a year ago or 2 years ago as the oldest team in baseball, I was actually very happy. I think wisdom and experience has something going for it. Your SAB is fantastic. I mean, your board is fantastic. You've got a lot of people here with a lot of experience. Just a couple of questions, more so just from a granular perspective. One is, will you qualify for a PRV, priority review voucher for Niemann-Pick C, assuming that gets approved because as a pediatric disease, or not really?
No, that's a great question, Hartaj. Yes, the answer is we check all of the boxes as being first approval in an active ingredient, and we do qualify for the PRV. So thank you for pointing that out. Yes.
Great. My other question is that, just going back to Sharon's point on cyclodextrins and the identification by, I believe, Dr. Liu, and then also the publication by the other physician mentioned on the SAB. Sharon, I guess, how come you chose to go through the path of a startup? And I get it. I understand the upside potential, everything there. But how about, why not try to see if a larger company would take this on and try to push it through as opposed to going through it with the route of Cyclo Therapeutics?
I think Scott should take that one.
Scott, if you,
Hartaj, you mean, partnership is what you're talking about.
Partnership, or somebody who's kicked the tires and said, all right, we can help with running a large program. Alzheimer's, I imagine the program is not going to be small, right?
No. So I'll try to answer this as best I can. We are very, look, I've been an investment banker for 35 years. We've got a very experienced strategic board of directors. We understand our responsibility to our stakeholders, and we very much are aligned and are in tune with what potentially could be partnership opportunities for our company. I probably think, Hartaj, that's as far as I should go right now. But for sure, we understand the value of that, which is, frankly, it's one of the reasons why, again, if you look at our management team, bringing Jerry Cox in, which was substantial add to our management team from the standpoint of, not only from getting to understanding clarity on the science and validation of the science, but also looking at those types of companies that, might have an appetite to look to partner with a company like ours, not only for NPC, but also for Alzheimer's.
Yep. And then also, I guess my other question is, you're using these molecules, cyclodextrins, to essentially trap cholesterol, trap tau. Is there, how to think about kind of, and I imagine you've done the work here, Sharon, but just in terms of how to think about safety, I mean, what dose level can you get up to? What does that sort of just how to think about that, because your approach seems to be fairly new relative to other companies that are using biologics, et cetera. But how to think about safety and being able to push the dosage with these molecules that you're utilizing?
Well, that's a great question, Hartaj. Thank you for that. What we've done, first of all, we published a paper of 12 case studies, and this was when very little was known about cyclodextrins and NPC, and individual physicians did push the dose, and they learned a lot about what would be tolerated well. The highest dose used intravenously was 2,900 mg per kg, and these are large doses. In our formal trials, phase 1 and phase 2, we used up to 2,500 mg per kg, and very importantly, the safety profile for those doses, or for that dose, is very good. Even at that, what you would think of as a very high level. What we're very interested in is that the drug crosses the blood-brain barrier or gets into the cerebrospinal fluid, whether it's through the choroid plexus or some other. It gets into the CSF. So how much drug is available for CNS? These are questions that these are points that we're very interested in, because we see benefit clinically. And we are matching that to the drug being present in the CSF, and therefore touching the neurons and the neuropil in the brain. So that's a sort of that's what we can say about dose level.
Yep, no, that makes sense. The other question I would have, Sharon, is, and I'm not as familiar with Alzheimer's, but in covering companies that are focused on neurological disorders, we hear, the scientists at these companies say, for example, that, a lot of neurological diseases are not just a disease of the brain. I mean, that's where a lot of, human thinking goes on. So you kind of see it from that level, but they're also disease of the peripheral nervous system, right? So you'll be able to get into deep brain tissue, where crossing that blood-brain barrier is very important, but then be able to also influence the peripheral part of the nervous system. How do you think about that when you think about Niemann-Pick and Alzheimer's from your approach of using these cyclodextrins?
Well, that's a great question. For me, the common denominator is cholesterol. So we have to think about what is cholesterol doing in neurons in the brain, or even at the neuromuscular junction. And I think that a part of the story that is certainly becoming more apparent is that cholesterol plays a very important role in lipid rafts and in parts of cellular membranes where cholesterol is accumulated. And in those lipid rafts, there are proteins embedded. So whether it's proteins involved in transmission at the neuromuscular junction, or whether it's proteins, say, involved in the creation of amyloid, which is the case in central neurons. We're very excited about looking at our drug through the lens of cholesterol, because we know that it interacts with cholesterol, and looking at mechanisms of action that will sort of tie up that kind of story.
Yeah, so just so I'm clear, it's actually going after cholesterol and being able to, is it literally just see the clearance both in the deep brain structures and the peripheral structures, that you can get a sense that your drug is actually working on both those levels? Is that?
Well, we haven't measured that, but I do think that there's a lot of, with the wave of momentum around Alzheimer's now, and other neurodegenerative diseases, we're going to be learning a lot as a community about the role of cholesterol, I think, in underpinning many of these diseases.
Understood. No, that's really, really fascinating. The other question is, once your pivotal reads out in NPC, Sharon, what's the sort of next step? I mean, have the regulators given you enough confidence that you can file potentially on one trial, or do you think you'd have to run another one? Is there any long-term safety follow-up or anything else that might be needed with the NPC trial?
Sure. Well, what we have in our pivotal right now is an interim analysis at 48 weeks, and we are building our trial such that if we do see efficacy at the interim analysis, we will be able to move forward with the regulators. We'll have discussions with the regulators about what that actually means. If we do see efficacy at the 48-week trial, all patients will move into open label. That's already built into our protocol, which is on clinicaltrials.gov. So we're planning out our open label already as part of our larger platform.
Got it. And Sharon, usually when an interim happens, the statistical bar to hit it is a lot higher, especially if it's one trial. Is it just safe to assume that is that the way to think about this pivotal also? Just if you do hit that interim, that bar is going to be pretty high in order to be able to cross it.
Well, we are working with Worldwide on our statistical sort of analysis, and we do believe that our construct is such that if we do give up some of our alpha for our p-value, we'll be able to reach our bar. And if we don't happen to see efficacy, then we will be able to reach it at the end of the trial.
At the end of the trial, yeah. Yeah, but I guess what I'm trying to say is that, usually like, let's say if the p-value is 0.05, then for an interim, it's like 0.005, right? It's a small portion of that 0.05. And I'm not saying those exact numbers, but is that a logical way to think about it for the interim also?
It is. And we haven't published all the fine points of our statistical analysis plan, but one does give up something during an interim analysis.
Yep, got it. Got it. No, if you hit it, that would be, I think that would actually give, the markets, investors, patients, physicians, a lot of confidence. That's a very high bar to cross. And then just in terms of like your Alzheimer's project, there's so much controversy in Alzheimer's right now, rightfully or wrongfully, I don't know. That's way above my pay grade. And I know one of my colleagues covers Biogen, so I'll leave that discussion up to him. But I guess, does that, Scott, how is that controversy adding or subtracting to your approach? I mean, number one, are the regulators under more pressure now to make sure trials are done in a specific manner, specific whatever that means, or more typical of clinical development programs, as opposed to, some therapies that were approved recently. And number two, just from terms of investors, do you see a greater sort of interest in Alzheimer's now that a drug has been approved, in this area, for Biogen, or do you feel like the controversy is more for drag, when it comes to this space?
Well, I'll answer first, and then maybe Sharon can talk. Hartaj, we've not seen controversy. We of course know that the Biogen approval has brought a greater light and a greater expectation to put pressure on regulators to approve other drugs. We're hearing interesting things about Lilly as a point of fact, where Lilly is going to aggressively go after the FDA, similar to what Biogen is doing or did. But for us, I think what's really key is that driving our science and driving the opportunity, and frankly, I think the marketplace is giving us. Well, I don't think they're giving us much credit for our Niemann-Pick C asset, to be frank, Hartaj. But I don't think they're even attributing anything to Alzheimer's. I think once we know our outcome of our meeting with the FDA in this last half of the year, and if FDA does say to us, yes, you can move forward with the phase 2B program, then I think the marketplace, I think they should begin to pay attention, because it's obviously, as you know, a much further along program with greater expectations. I will say, look, it's an N of 1, but you have to start somewhere. And backed with the Flint-Beal data that was done in the mouse model subcutaneously, we're quite excited about our Alzheimer's program being a true potential treatment modality, and moving it forward with clinical programs.
Scott, last question on Alzheimer's. If the FDA was to allow you to move forward with phase 2B, what would the contours of that look like? I mean, we're talking a few hundred patients, high hundreds, and the length of how long would the study have to be roughly? Phase 2B like that?
Do you want to take some guesstimates at that?
Sure. Well, we haven't disclosed the parameters of that particular trial just yet, but you can imagine that, a phase 2 trial would be on the order of maybe less than 100 patients. That would be normal. It would be normal parameters for a phase 2 trial, including in duration.
Yeah.
Understood. Got it. Well, I can see why, Scott, you are so frustrated with your market cap, and Sharon, now I know why you left the NIH to join this company. So
good analogy.
Really good, exciting days ahead. Knock on wood. I'm keeping my fingers crossed for Cyclo, and we look forward to keeping the conversation going.
Great. Thank you, Hartaj, and thank you, Oppenheimer, for letting us present today. Truly.
Thank you.
Thank you, Scott. Thanks, Sharon.
Take care.
Stay safe, everyone. All right.
Yeah.
Bye-bye.